Good morning, good afternoon, and welcome everyone to today's live broadcast, Lifting the Veil on Innovations and Product Launches 2020. I'm Alexis Krauss of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is brought to you by LabRoots and sponsored by Bruker Daltonik. For more information about our sponsor, please visit their website at bruker.com. So, let's get started. I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. To do so, simply type them into the Ask a Question box and click on the Send button. If you have trouble seeing or hearing the presentation, click on the Support tab found at the top right of the presentation window, or report your problem by clicking on the Ask a Question box located on the far left of your screen. I'd like to now introduce our presenter, Dr. Markus Kostrzewa, Director, Senior Vice President R&D, Regulatory and Scientific Affairs, Bruker Daltonik Microbiology and Diagnostics. For a complete biography on our presenter, please visit the Biography tab at the top of your screen. Dr. Kostrzewa, you may now begin your presentation.
Welcome, ladies and gentlemen, to the presentation about the innovations of Bruker in the area of microbiology and diagnostics. Most of you may know me. I'm Markus Kostrzewa. I'm heading the innovations part of microbiology and diagnostics of Bruker. And I'm happy to present to you now what are our new developments, our developments and research results in the year 2020. Normally I present this in seminars around the ECCMID and ASM. This year, unfortunately, we were not able to do that. So we decided for this online format, and I hope it will be informative and you will enjoy it. My presentation will have 4 parts. In the first part, I will guide you through some developments and some state-of-the-art in microorganism identification using the MALDI Biotyper and developments we did regarding this. The second one will be about rapid antibiotic susceptibility testing using MALDI Biotyper. Then I will guide you through some new developments in hospital hygiene and classification using the IR Biotyper. And last but not least, I will give you a short preview about developments in molecular diagnostics. MALDI Biotyper Series 1 is our youngest member of the MALDI Biotyper family that we are presenting this year. Essentially, it's something like the little brother of MALDI Biotyper series, and the specialty in this is it has only the positive ion mode to cover all the routine workflows you have in your laboratory. In other cases, it's just very similar to MALDI Biotyper series, as you know it, having a smart beam laser with 200 Hz for fast measurement, having a high capacity vacuum system for rapid accessibility for measurement. The electronics is new and environmentally friendly. We, in addition, improved the usability by improving the outer design for better disinfection. And what's also a nice feature is that the system status can be checked via an LED strip. In addition to development on the hardware, we also expanded the capabilities of our in vitro diagnostic software for the European Union certification CE. We expanded the IVD software by adding a software module for subtyping. So what does subtyping mean? Subtyping in this case means you can identify markers, as an example for resistance detection of bacteria, or you can sub-differentiate species or subspecies which are closely related and cannot be differentiated by the standard workflow. Now, as first applications, we have introduced the differentiation of CFIA positive Bacteroides fragilis from those that are CFI negative. The difference is that the positive Bacteroides fragilis are carrying a carbapenem gene, which makes it highly resistant against carbapenem antibiotics. The second functionality is the detection of Black KPC expressing Klebsiella pneumoniae and Escherichia coli, which also are having a strong resistance against carbapenem antibiotics. As a third, we introduced the first species differentiation, which cannot be achieved in a simple way. In this case, the differentiation between Mycobacterium chimerae from Mycobacterium intracellular. An important differentiation, as in the recent years, Mycobacterium chimerae has been detected from heater cooler systems infecting people. So here you can see how the output of such a detection looks like in a further column now. In addition to the identification, the log score, and the comments, the subtype is given as CFIA positive or negative, KPC positive if applicable, or typed as Mycobacterium chimerae or intracellular as it applies. A very important part of MALDI Biotyper, of course, are its databases. The main databases, the large RUO and IVD-CE labeled databases, have been extended significantly, I would say. More than 1,000 new reference entries have been added, representing 21 genera and 278 new species. With this, we're further extending the clinical coverage of diversity, but also of species. Here are some species given that have been added in addition. I might point on additional strains also for Staphylococcus argenteus and Staphylococcus schweizeri, which now can be validly and well differentiated from Staphylococcus aureus. In addition, we added also not only clinical but environmental species. And another important feature is we improved and renewed the taxonomy. So taxonomy is permanently changed, and we are doing our best to being up to date with our databases each year. I would like to give you some examples for the improvements that we have achieved. And I have to say, this is very important for us that you help us with this, and that is a very good collaboration with our users and customers, that we get input from you and that we get also recommendations from you how to update, how to extend, and how to improve our database. One of these hints came in the area of Acinetobacter genus. There were several publications that said Acinetobacter genus is not good enough covered by databases. It's more diverse, there are more species, and it could be improved. We listened to that, we saw that in publications, and we also discussed this with our users. And finally, we received a big number of Acinetobacter strains, explicitly from Alexander Nemec from Prague. We evaluated these strains, could incorporate 169 of them into our library, now having a huge extension overall with 22 new species. So an enormous coverage, and you see how we can extend our database in the interaction with you. A very special database is the one for the MALDI Biotyper CA, or clinical application. This database and the system for it are only for sale in the United States and Puerto Rico, as the regulations of the FDA are different from those regulations where the IVD-CE labeling can be applied. We have now also improved significantly the database for the MALDI Biotyper CA system. We have added 59 new species and groups, and also we added a renaming, an improvement of naming and up-to-date naming for a lot of species and species groups. Also here I want to give you some examples. Very important is the genus of Elizabeth Kingia. This is something where we can see the historical development in taxonomy and also the influence it has on clinical diagnostics. Initially, Elizabeth Kingia, Bricola, and Meningoseptica have been defined in 2005. Just in 2011, a new species, Elizabeth Kingia anophelis, has been defined and determined. So later on, it appeared that this is the main species, and formerly was just classified as one of the other two species. We have investigated that thoroughly, added a number of strains and cover now this species diversity and species differentiation very well, and have them in the MALDI Biotyper CA database, inclusive very thorough validation. Also for Acinetobacter, we added a further species, which is now part of the Acinetobacter baumannii complex. This is formerly called Acinetobacter Yeaxhorniae, which was found at the end to be the same like Acinetobacter lactucae that has been defined earlier, and also now is part of the complex and can be nicely differentiated from the other species. And as a last, something special I would like to point on, Wolfratimonas chitinoclastica. This species was the one that was the first that we had on our t-shirt in ECCMID, where we sometimes present a t-shirt. Somebody has found something really rare with the MALDI Biotyper, and we call it is the species of the year, and say, what have you identified today? So this was the first example detected by a German customer. Later on, once we had it in the database, it was detected again and again, especially by clinical laboratories, especially from blood cultures. So we collected these strains. We even found strains in our earlier clinical studies in the United States for MALDI Biotyper CA. In the pre-identification, wrongly identified as another species by other systems. Now we have collected enough of them to add them to the MALDI Biotyper CA database and do a thorough validation according to the requirements of the FDA. So this is not something esoteric. It's found again and again, and it's of clear clinical relevance. So you see how things are changing from esoteric to clinical relevant bacteria. Maybe for most of you, the biggest surprise in our development today is that we are moving towards automation of sample preparation for the MALDI Biotyper. Here you see the soon coming MALDI Biotyper Pathfinder equipped with a feeder solution. The MALDI Biotyper Pathfinder is dedicated to the automated preparation of colonies for bacteria and yeast from an agar place onto the MALDI target, including the matrix deposition. The feeder option is for the automated feeding of the Pathfinder from a hotel, and the system has a nice traceability and documentation capabilities, as I will show you. Here the workflow of the system Pathfinder, including the feeder. Once you start a project and you run, of course the MALDI plate is placed into the system and all the reagents. After that, after a start, the automated scanning starts of the Petri dish, a barcode that is at the side of the Petri dish, registration of the sample. The sample is automatically inserted in the dish, in the system. So this part is the connection between feeder and Pathfinder. Then an optical scan of the plate is done. A colony is selected. Thereby, what's happening is that the Pathfinder makes a recommendation for a colony, and you can accept that, or you can select manually another one. After this selection, first on the target, formic acid is deposited, and after this disposition of formic acid, that's something special, the colony is transferred into this droplet of formic acid. After drying of the sample, matrix is automatically deposited. After that has been done for all samples, the run is closed and finished. The sample can be introduced with the target plate into the MALDI Biotyper for analysis. So overall, the system shows nice features like a lot of cleaning and sterilization processes. It cleans the needles automatically. It also prevents crystallization of matrix in the needles, and the wires are sterilized that are transferring after the transfer by heat. The reagents are dosed by separate needle, formic acid and multimatrix. It's contactless. We don't use pipette tips. Colony and selection, as said, can be automated, but also can be manually changed. And there is a special function for detection if bacteria are slimy and would cause trouble by transfer. Then the process is interrupted to prevent any contamination inside the system. The full workflow is transparent and traced very well. There are a lot of pictures made and stored from all process steps, so later on for troubleshooting it's very easy to follow the whole process. Barcodes are used for traceability, targets automatically recognized, and also the petri dish surface, the heating, the drying limits are all automated. So far about the identification using the MALDI Biotyper and the sample preparation for it. In the second part, I would like to guide you through our developments according to rapid antibiotic susceptibility testing using the MALDI Biotyper. This is an overview about the workflow we call MBT Fast Forward. What does that mean? We have a workflow covering two aspects. One is the rapid detection of antibiotic resistance or susceptibility using MALDI-TOF, namely the MALDI Biotyper, as the readout. This part works in 5.5 hours plus preparation time, so same-day results are available. As a second, we have an overnight incubation of the same sample to get a real MIC determination at the next day. We are using combined antibiotic plates, like here shown in the lower part on the left side. The first two rows you see are for the fast part, same-day breakpoint detection. The second part, forward, is on the right side here, where you see many dilution points for more antibiotics. The whole process is near to complete in development. I will guide you through the results of our developments. We have, according to consumables, you have a software module and also a tool for incubation and removal of supernatant. Here, a quick view again on the workflow. You see the bacterial sample coming from an agar plate or from the pellet of the sepsi typer, so from the blood culture isolation, is put as an inoculum in an antibiotic plate, and now a part of this plate is used for the fast workflow. This means at the same day you get a result by inoculating from there a MALDI Biotyper target plate, incubating that in a special chamber. When the bacteria are resistant, they will grow in a droplet on the target, and when they grow, they will adhere to the MALDI target, and later on you can measure a spectrum and identify. A susceptible bacterium will not grow, and there will be no measurement of a spectrum with the bacterium, and there will be no identification. So same-day result for urgency ST result. On the other hand, the same sample inoculated in a classical broth microdilution plate, and this is incubated overnight, and then the next day there is a real MIC detection, there is a confirmation or correction of the MBT FAST result, and there are more antibiotics which are determined. So this system will come with some consumables that are supporting the whole workflow. One of those is of course the antibiotic plate. Here our Bruker Merlin branch is supplying us with these according plates and antibiotics. They have a lot of experience in this and are known for high quality broth microbiology dilution assays. It will contain 9 antibiotics for the rapid AST, same day result with breakpoints according to UCAS. It will have 16 antibiotics overall for next day real MIC determining. The second consumable is a special matrix that we supply. That's a matrix containing an internal standard which ensures validity of analysis. Because if no bacterium is grown and no bacterial spectrum is measured, nevertheless, the standard has to be measured. If the standard's there, then the analysis has worked and the bacteria are not grown. If the standard is not detectable, something else was wrong. For the removal of the supernatant after the incubation, we have some pads which are removing the supernatant together with a stamp in a very rapid and very simple way. And at last, we have some special broth for the growth in a MALDI-TOF compatible way. This is a view on the incubation chamber, which we are calling MBT FAST Shuttle. That's a multifunctional hardware dedicated to MBT FAST, but also potentially for other applications. It has a touchpad function, it's very dense, it's very controlled, easy to use, and also just small, fitting on each workbench. An according stand for the removal of the culture supernatant after incubation is also supplied. You see it here on the right part. And besides the application for MBT FAST, this tool also will be developed further to enable a rapid drying for the ID workflow. I will show you later, but also for other assays like a fast assay to confirm pneumococci, and for acceleration also and simplification of our STAR-BL assays. Also, a part of software, of course, is needed for the analysis, for the measurement and analysis of according spectra to MBT FAST. You see a view on the GUI here. It's a software module for MALDI Biotyper Compass dedicated for this analysis. The whole workflow is software guided. Measurement is done and started automatically. Also, the sensible intermediate resistant classification is done automatically. There is a plausibility check and warnings of inconsistencies. The system gives also a PDF report, and data can be exported via comma-separated values. As already mentioned, this shuttle will not be only applicable for MBT FAST AST, but also for other workflows. And the most simple workflow for that is the acceleration and standardization of the drying procedure for identification. We have observed in some laboratories that in some very humid summers, as an example, the samples are drying slowly, which also can have a negative influence on the later on measurement. Using the MBT shuttle, this all can be accelerated and standardized. We have seen and measured acceleration of the whole workflow from starting the preparation to the measurement of up to 50%. And the nice thing is, up to now 10 different species have been thoroughly tested. The log scores of the quality of identification we get is at least as good as in the normal procedure using room temperature, maybe even somewhat better. The influence of environmental conditions is clearly diminished, and the system will be validated for MSP 96 polystyrene target plates, but also for biotargets, of course. Further possible workflows for the MBT FAST shuttle are the acceleration and simplification of our STAR-BL workflows for detection of carbapenomases and for detection of cephalosporinases. As you've seen here, we also think that we can reduce time to the result by half approximately. And a further test is published by Evgeny Idelevich in just this year. That's the confirmation of pneumococci by an objective automated biosolubility test using MALDI-TOF MS, very similar in the procedure to MBT FAST for AST. We developed this as a standardized assay using these tools, and it can be used later on for same-day results for confirmation of pneumococci after identification with the MALDI Biotyper. So far about rapid AST using MALDI Biotyper and the tools and consumables around that. In the third part, I would like to guide you through our improvements and novel developments for hospital hygiene, subtyping and classification of microorganisms using infrared spectroscopy, namely the IR Biotyper. You see here the picture of the IR Biotyper we introduced just recently. We are now already in the third version of the software that we've developed with a lot of feedback from our users and own ideas. So, coming from the new interface, you can see here some improvements like increased robustness of the system after some experience in the field, improved usability in the usage of the software, especially important, the introduction of a new workflow in parallel to the classical hospital hygiene workflow. That is the implementation of building of classifiers, of machine learning to create classifiers and apply classifiers. I will guide you through this later. We extended the wavelength that can be analyzed to the full range that is measured. When we started, we restricted ourselves to the carbohydrate range, which is most informative, but there's also information in other molecules, and we have expanded the system that can be analyzed now. We also improved the visualization, namely by introduction of a nice 3D scatter plot. And also very important, we included some new connectivity. On one hand, the MALDI Biotyper can be connected to the IR Biotyper, as it should be, as members of a family. So the species identification generated by MALDI Biotyper can be transferred directly to the IR Biotyper, and in future these capabilities of transfer will be expanded. On the other hand, IR Biotyper also can be connected to the laboratory information system and transfer data there. So at first, from the application side, I would like to show you some nice result of the, now let's call it classical workflow of the infrared biotyper for real-time epidemiology in hospital hygiene. So this example comes from Bologna, from the university hospital there, a quite big university hospital. And the work was performed, and I think that is really remarkable, during the COVID crisis in April, when, as you know, in April, the hospitals were full, especially university hospitals, and a lot of urgency was there, and the people were really under stress, so it was hard to do anything else than COVID testing that time. Nevertheless, this hospital hygiene observation was made. It started by an outbreak of an Acinetobacter baumannii that was carbapenem resistant, only colistin susceptible, in a ward of the university hospital. And you see here the first measurement. So there was an index patient, patient 0, and after that 8 more samples have been taken in the first week after this first patient, and they all clustered closely, and I would not like to say nicely, together with the index patient. The negative control is clearly unrelated, and you see also it's totally separated. The threshold that is applied here is as we give it in our manual at 0.4. So normally now each day a sample would have been measured, but as this was in the pandemic situation in very bad conditions, much worse than we have it today, and we all hope it will not come back again. So in this case, only once a week the measurement was performed, which nevertheless was remarkable because of the shortage of personnel. You see here now the result after the second week. So at one hand, again, 8 new patients appeared. So it spread over the ward. And in addition, other samples from another hospital, another ward and another hospital, you see in the lower part, given here in violet, were also separated from those, but also clustered. In that pointing already on, there might be another outbreak in another hospital. 1 week further, again, more patients got this Acinetobacter baumannii on the initial ward, as you see, all gray. So it's now 24 patients there. And in addition, on a further ward in a further hospital, the strain that caused the second outbreak, you see it below, this dark one among the violet ones. This one clusters together with the violet hospital, let's call. So you could say, now, well, maybe that's random. But actually what had happened is that the patient in this third hospital, third ward, came from this lower hospital, from the hospital where the second outbreak happened, and transported, or hopefully not seeded, this multi-resistant Acinetobacter baumannii again in a new hospital on a new ward. I think a very impressive example how the IR Biotyper can be seen as a method to observe and hopefully prevent and reduce the spread of an outbreak at a hospital ward. Coming to some more features of the IR Biotyper 3, here you see a 3D scatter plot, in this case after a linear discriminant analysis of serogroups of Salmonella. Salmonella is an area where we're currently working hard on. It's very fruitful to work on this. With the IR Biotyper, we get a very nice separation from the serogroups, and that's also depicted here. You see here the different colors that are representing different serogroups different O serogroups of Salmonella. And interestingly, when we reduce the number of different serogroups and focus especially on this O9 group, then you can see that inside the O9 serogroup, Salmonella typhi, which is highly pathogenic, can be well separated as a subclass in the group. This has been now confirmed by a lot of strains, and we're working with our partners in Bologna on that, also for our publication. So such results now enable us to further develop it from a just observation by similarity to classification. What I've shown you up to now was unsupervised analysis without information what is related to what, just looking at the similarity of strains, and thereby, by similarity, detecting something like an outbreak, tracing such outbreaks, tracing also contaminations in pharma companies or in food industry. On the other hand, if we know classes that can be differentiated, and we see that such classes can be separated, like as an example, the serogroups of Salmonella, we now can use machine learning to calculate classifiers, mathematical biomarker-based classifiers that later on can be used for classification or subtyping or differentiation. So the workflow is from a training data set that is well defined of node samples, spectra are acquired. These are fed in the algorithm, the machine learning algorithm, in our case now an artificial neural network. This uses it to learn how to separate the different classes, and then creating a model, a so-called model, classification model. This classification model later on then can be used to classify unknown samples, and the output is, as you see here, then similar to the MALDI Biotyper with a green and yellow and red quality, with the classification and detailed information. So we have applied this as an example in some approaches now, as an example to Salmonella, but by the way also for pneumococcal serotyping and for the differentiation of Legionella serogroup 1 from the other serotypes. So here you see an example for Salmonella, differentiation of different serogroups, and the result of those. 23 serogroups were used in this case. A training set measured on a TSA agar was used to create the training set. From this, with the neural network, a classifier could be trained and created. And then 166 independent isolates were analyzed. And these were grown from TSA, but in addition, even a larger number of strains was grown on another agar, on Columbia blood agar, and also tested against this TSA-derived neural network. And as you can see, with the TSA medium, very good results could be obtained. There was no misclassification in this test, but there were some not classified. That means red. But interestingly, also somewhat worse even from Columbia blood agar. And normally you always try to be as standardized as possible. Even from this, more than 80% of the samples were correctly classified against the neural network created based on strains grown on TSA. Only 4 misclassifications happened. So you see there's quite some robustness in this method, especially for Salmonella. In addition, also, of course, we looked at the TÜFI problem. So we created a subnet, a network that was just applied against the O9 determined strains. And indeed, we could see with this network, the non-TÜFI from TÜFI Salmonella could be differentiated. This was done in collaboration with Simona Ambretti from the University Hospital of Bologna, but also with a reference center for veterinary and food from Parma, Stefano Pongioli. So also a nice collaboration of our customers and partners that are experts in their field and then can supply the expertise about samples and of course also the samples. So far about hospital hygiene IR Biotyper. At last but not least, I would like to give you a quick view in our molecular diagnostic branch. We are also now in the fight against COVID, namely in the diagnostics of SARS-CoV-2. So, as a series of developments for respiratory assays, at first we have developed a real-time PCR assay that can be used to identify with high sensitivity and high specificity the SARS-CoV-2 virus. It's called SARS-CoV-2 Plus because it also can detect the endemic human coronaviruses that are not as highly pathogenic. And as you see here from the analytical and clinical performance data of this CE-IVD marked assay, there's a very good sensitivity analytically, but also sensitivity and specificity are very good for this assay. And that has been also now confirmed. We have launched the system in May, and the feedback is very good for it. We get now to winter, and we all know, and we see it already, winter will get more problematic again. In addition to SARS-CoV-2 rising probably, because we're all more inside and close together, the flu might come back, and also other respiratory viruses. So, as an answer to that, we are developing an assay that can detect the SARS-CoV-2 virus, again with two different genes that are detected, which increases its robustness. But also it can detect influenza A and influenza B separately. It is also made to detect RSV A and B, not differentiated, but just as a warning. And all this is in a 5-color single tube assay. Currently, we have this system as an REO kit, and the CE labeling is in preparation. Validation studies are running and look very promising. We intend to launch the system for the detection of these different viruses during the next weeks. And at last, we are also in development of a broader panel, of a real broad panel for viruses, bacteria, and fungi that can cause respiratory infections, the so-called Syndromic panel. This we do with our patented so-called liquid array technology, and thereby it's possible to have in only 2-tube assay 17 different respiratory viruses, 9 bacteria, and 1 fungus. This all runs in 2 tubes and enables such a very broad detection as normally only possible with systems that are used point of care for few samples and thereby not in a higher throughput. This is in development, and we are intending to launch that in the way of 2021. This is one of the assays that we are developing, which are called so-called syndromic panels. That means detection not only from one class like virus or bacteria or parasites or fungi, but just according to the syndrome, wherever it comes from, detect the pathogen. Further developments in this area are, as an example, detection of pathogens that are causing gastrointestinal infections. And another one, the first one, has been already developed and is IVD-CA labeled, and that is for sexually transmitted diseases. It's an assay, and you see the analytical and diagnostic values of this sample from the IVD-CA labeling here. It's done to detect 7 different pathogens, partially with dual targets. It's a single tube assay, also using the liquid array technology, and by that also enables a high throughput rapid detection of these pathogens in a simple workflow. This is just the start of development of such assays, rapid assays in high throughput that can be used to detect a lot of different pathogens or also mutations in the sample. So with this, I want to conclude my presentation, just pointing on other presentations that will follow, other webinars. There will be dedicated webinars explaining more in detail what we are doing and what we are presenting, about molecular diagnostics, about the sirius and related developments, about anaerobe susceptibility testing with the Moylan plates, about the MALDI Biotyper, and also about MALDI Biotyper sepsityper, where an expert insight will be given. I hope you enjoyed a little my talk and I could give you some interesting information. And by that, I would conclude with my presentation. And of course, I'm open for any questions. Thank you a lot for your patience.
Thank you, Dr. Kostrzewa, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. It will answer as many of your questions as we have time for. So let's get started. Our first question is, which additional sample preparation or tools are needed to get MBT subtyping IVD results? It looks like we might be having a little bit of technical difficulties with Dr. Kostrzewa, so we'll try to get him back on the phone.
Hello?
Hi,
Can you hear?
How are you? We can hear you, yes.
Yeah, sorry, obviously a technical problem. So I got the question.
Okay.
I got the question and I answered already, but I will repeat, of course. Actually, it's just a bioinformatics capability for the subtyping. So nothing is necessary from the sample preparation. It's just a method by bioinformatics, by another complementary method, to draw out more information of the same spectra that have been acquired for identification.
Perfect. Thank you so much for that. So we'll move on to our next question, which is it possible to replace or at least guide the selection of whole genome sequencing usage by the IR Biotyper in outbreak studies in clinical hygiene?
This indeed is also a very interesting question, and of course, over time we have to find out about the positioning of the system. We don't think and intend to substitute a gold standard like whole genome sequencing, but because of the high quality, but especially also rapidity and low costs of the system, we see it as a method to have a rapid answer, to guide and then be confirmed by other methods like whole genome sequencing. So I think it will be more a substitute for many analysis, and it will reduce the high cost, high effort analysis to a minimum where it's really needed.
Wonderful, thank you. Now, Dr. Kostrzewa, do we have to buy media from Bruker in order to have the barcodes?
No, Bruker will not sell media. We are not intending to go to this business. There you are free what you're selecting.
Okay. So our next question is, can the MBT shuttle be used for drying all, including ID-related MALDI targets?
Indeed, this is one of the aims we have. It's not yet validated for in vitro diagnostics, but we have tested quite a number of samples already with very promising results. And one of the intentions is indeed as the lowest effort application to use it to accelerate and also to standardize the drying process before identification.
Now, does this work with molds as well?
Well, that is not yet tested. We will have to have a look at that, but it might be that the micelle is not really suited for such a kind of preparation.
Okay. So it looks like this question, looks like our next question has a couple parts to it. So for the Fluoro type SARS-CoV-2+, are you using GXT-NA extraction carriages? If yes, what are you using for the amplification? And the fluorocycler or something else? And is the Fluoro type SARS-CoV-2+ an amplification kit? Okay,
indeed a complex question.
There's a couple parts to it.
Yes, and I try to explain that in detail. So it is an amplification kit, a PCR kit, which is validated indeed with a gene extract NA kit. In addition, we do bridging studies also to other extraction methods. So it's not only restricted to the cartridges for the NA kit, but it's validated based on that. Then it was about the PCR cycler that can be used. Indeed, the validation was, of course, done with the Fluoro Cycler XT, but it also runs on a couple of other PCR machines, and that you can find also at the leaflet. The validation was done also that it can be applied as an in vitro diagnostic on that. Was there a further part of the question which I missed? I think its application kit is a PCR kit, of course. It's a kit for PCR amplification, a single tube for several targets.
Wonderful. Thank you so much. I know there were a couple parts to that question. So we'll move on to our next one. Are the subtyping results also contained in the LMS export or LIMS export? Excuse me.
The multi-parameter subtyping. Yes, indeed. That is also now part of the transport or the export to the laboratory information system.
Wonderful. Now, Dr. Kostrzewa, our next question. Are the subtyping results also contained in the, oh, excuse me, I just said that one. Well, our next question. With the FAST module comes the new HT platform. Do the non-HT MBT software and its workflows still run with this new FAST workflow?
Okay, I understand. Yes, indeed, we are moving our data analysis for MALDI Biotyper to the so-called HT or high throughput platform, and the MBT FAST module will be a part of that. But it's not a problem to have both systems, so the Compass software as well as the HT software on the same system, and thereby having all the capabilities that we are offering on the same system. Also, the identification and subtyping is performed and FAST is performed on the HT platform. So there's compatibility of both systems, and they can be run in parallel as long as we don't have all functionalities on the HT platform.
Wonderful. So we do have time for a few more questions, and we want to thank our audience for all these amazing questions that are coming in today. Just a reminder, all those questions that we do not have time for today and those that come in during the on-demand period will be addressed by Dr. Kostrzewa with the email address that you provided at the time of registration. So our next question, what is the capability of connection planned between MBT to IRBT?
Yeah, we did the first step now by delivering the species information to the IR Biotyper. So the species identified by the MALDI Biotyper is transferred. But of course, in future, we intend to expand that. And you just can imagine all the information that we are generating on the MALDI Biotyper side, which might be interesting. As an example, a resistance profile that is stored in the MALDI Biotyper server, or subtyping information. All this, of course, in future step-by-step can be transferred and connected. In a last step, I nicely can imagine that also some typing results obtained by MALDI, which is not the same but complementary to infrared typing, might also be combined, and thereby a kind of grid of different typing technologies with high resolution can be generated. So that's something over the next years we will introduce step by step.
Now, will the IR Biotyper work on mycobacteria?
That's a good question. Hopefully, but I have to admit that we did not test that yet. In principle, it should work, but it might need some special sample preparation for that.
Wonderful. Thank you so much. So, our next question, how long does it take for the MBT FAST give the AST result? You know, the current technology takes approximately 16 hours.
Yeah. So the incubation time for enterobacteria, that's the first assay we're working on, is 5.5 hours. We are somewhat limited here by the growth of the bacteria and the expression of their resistance. Including sample preparation, it will be approximately 6 hours. So sample preparation before and measurement. which is for those blood cultures that are positive in the morning, a same-day result at least. But I have to mention, of course, that the detailed result also will be then available next day on the forward part of the plate by classical broth microdilution, and then giving also a true MIC and also correcting if the short result has shown some inconsistencies.
Now, will the FT-SARS influenza assay also run on non-Bruker PCR machines?
Yes, indeed we are validating this also on other machines. There might be at least 2 further cyclers where it's running on. As this is a high complex multiplex, it has to be adapted to machines that are capable for the separation. But as said, in addition to the FlowCycler XT, at least 2 machines have been identified. They also, of course, will be mentioned in the package insert.
Great. Thank you so much for that clarification. Now, is it possible to replace or at least guide the selection of whole genome sequencing usage by the IR Biotyper in outbreak studies in clinical hygiene?
This is, at the end, the intention of what we want with the IR Biotyper. We think that whole genome sequencing or other high-resolution, high-effort technologies can be partially replaced, and thereby the throughput and speed can be in hospital hygiene can be clearly increased.
Wonderful. Now, about Pathfinder, can you compare the result of systems with manual sample preparation study?
We don't have yet validation data for that we can publish. You have to understand that we will wait until that has been finalized and we can IVD-CE label. But from the first results and the feedback of testing sites that we got, we can say that the results look like at least as good, if not better, as manual preparation. I think that especially in, let's say, in hectic laboratories, with also maybe some new technicians that are not as skilled, and the manual work will even be of lower quality as a standardized system like the Pathfinder, which at the end each day works the same level, and that's a quite high level as we have observed.
Now, regarding the IR Biotyper, you've already introduced some classifiers. Which additional ones can we expect in the near future?
Okay, about the classifiers. So currently we are working on a number of classifiers already. We have seen, as a very simple example, that the serogroup 1 from Legionella pneumophila can be nicely separated from the other serogroups, which has some meaning because of the higher infectivity. We have already published and work more on serotyping for pneumococci. And as I've shown, also the serogroup differentiation and even some more differentiation in Salmonella is something we are intending. But there might be also other examples where it can be applied. We thought about Haemophilus capsular types as an example, but that has to be shown at the end in experiments, and not everything will work. Another possibility might be, yet not deeply investigated, as an example, the differentiation of Klebsiella capsular types. So we have some first results about that, and we also know from publications that in Klebsiella the capsular types seem to be nicely separated, but that needs, of course, a lot of work. And what is limiting here sometimes is the alternative technologies that are available to compare with the IR Biotyper, because they are very often slow, expensive, and expert systems. But over time, I see that we will create a number of such applications and classifiers. And of course, also users can do that themselves in the local situation and create their own classifiers and apply that.
Wonderful. Now, Dr. Kostrzewa, we have time for just a couple more questions, maybe one or two. So we'll move on to our next one, which is what is the Pathfinder throughput and timing? So
the Pathfinder throughput is approximately a full target plate in an hour that can be prepared overall. So it's not mainly the speed that makes the Pathfinder, but the precision, the automation, and the very good documentation of the process, including photographs.
Wonderful. Thank you so much for that. So how does the user select the colonies, and how does the Pathfinder locate them?
So of course, the simplest way to locate colonies is by eye, but the Pathfinder equipped with a feeder also uses a machine learning algorithm to identify colonies, and these colonies then are proposed for picking. But if the user thinks that his eyes are better, or the machine learning, of course, is not always the optimal, he also can change that and select another colony if that's necessary.
Now, it looks like we have time for maybe one more question, and this is a two-part question. Does the machine learning of the new IRBT software is comparable to the identification in the MBT? And can it point out that the newly examined strain is the same of an outbreak previously analyzed?
Again, a good question. So I understood the first part, how the identification of the IR Biotyper, if that would be applied, compares to the MALDI Biotyper. And here I would say generally the for identification, MALDI Biotyper is the better system because it's very robust, and it also does not need any information about what kind of organism has been used. It's just looking for a similarity match, and very few influenced by any medium or temperature. So the overall robustness, I think, is just hard to beat. On the other hand, for some differentiations where the MALDI Biotyper is not suitable, not every information is hidden in the MALDI spectra. Also, some people would like to see that. In these cases, we might see that a kind of sub-differentiation identification is possible using infrared spectroscopy. The second part, could you repeat the second part of the question?
Of course. Can it point out that the newly examined strain is the same of an outbreak previously analyzed?
Oh, yeah. We're looking here at probabilities, and at the end, that's exactly the intention. If you remember the slide that I've shown about the outbreak in Bologna, this is exactly what was shown there, that there was an outbreak, and at one point even there was, for the second outbreak, a transfer of strain in a second hospital. So this tracking and tracing is possible here. With some limits, we don't know how long it is over time. Probably some controls have been also measured over time to see that the system still works in the same range. But overall, that is, of course, one of the intentions of the system.
Thank you again, Dr. Kostrzewa, for your presentation. Do you have any final comments for our audience?
Well, my final comment would be that we will be happy to answer any more detailed question, deeper question, also by the specialists of the projects. Everything that is asked here should get a thorough answer. And I also would like to point on the upcoming webinars where my colleagues, specialists in their fields, give insights to the areas where I just could point on and give you a first overview.
Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information that you provided at the time of registration. We would like to once again thank Dr. Kostrzewa for his time today and his important research. We would also like to thank LabRoots and our sponsor, Bruker Daltonik, for underwriting today's educational webcast. You can view the webinar on demand. LabRoots will alert you via email when it's available for replay. Please join us next week for part 2 of our webinar series, Fluoro Type Technology as Reliable Diagnostic Tool During COVID-19 and Flu Season. That's all for now. Thank you so much for joining us, and until next time, bye-bye everyone.